Hello, everyone, and welcome to Talk Nerdy. Today is Monday, July 22nd, 2019, and I'm your host, Cara Santa Maria, and today we're going to be chatting with Dr. Kevin Holden, and we will get back to Kevin in just a minute, but first I want to thank those of you who have made Talk Nerdy possible this week. Remember, Talk Nerdy is and will always be 100% free to download, and that's because we rely on the support of listeners just like you. So if you go to patreon.com/talknerdy, you can pledge your support just like Phil T-Bear, the Zombie Drummer, David J.E. Smith, Gabriel, Felipe Jaramillo Gonzalez, Brian Holden, and Jeffrey Sewell do each and every week. All right, guys, let's dive into the show. So, like I said, this week we're going to be talking with Dr. Kevin Holden. Now, Kevin this is such a great title. He's the head of science at Synthego Corporation. And Synthego is a genome engineering innovation company. We're going to talk a lot about CRISPR. He's going to explain all about what that means. So I guess without any further ado, here he is, Dr. Kevin Holden. Well, Kevin, thank you so much for joining me today.
Hi, Cara. Yeah, it's great to be here. Thanks.
Of course. So we are going to be talking about all sorts of cool stuff that I'm not sure that I've talked about too much on the show before. Anybody who's listening to Talk Nerdy, who's like a regular listener, knows that I also work on another podcast called The Skeptic's Guide to the Universe. And I think one of our favorite topics on SGU, almost anytime there's like a big relevant news story that comes out, is CRISPR-Cas9, gene editing, kind of the more modern approaches to gene editing. And so I think it's going to be really cool to do more of a deep dive today on the podcast. So Kevin, you are the head of science. Is that your title, head of science at Synthego?
It is, yeah. That's the cool title, right?
That's a really cool title. That's amazing. So being the head of science at Synthego, that means that you are a scientist, and you were a scientist before you started working there. Knowing that you work in CRISPR-Cas now 10, or Cas, is it Cas10 or CasX?
Most people calling it CasX, it seems like.
CasX. Okay, cool. In CRISPR-Cas9, which we'll get to and we'll really start to dive deep into, is your background in genetics? Is it in microbiology? Is it in another type of science that has some crossover? It's so multidisciplinary now. What did you actually study in school?
Yeah, so I guess just to refine on the title part a little bit. The company that I work for now, Synthego, we have lots of engineers and chemists and other people. So we have science people and engineering people. So I get to be the leader of the science. So that's kind of cool.
That's really cool. It's kind of like how JPL is down the street from me here. There's a lot of scientists, but even more engineers doing all this cool stuff at NASA, and they do kind of keep those. I mean, they work together all the time, but they keep those teams pretty separate in terms of the, I guess, management approach to it. But like at JPL, at NASA, there's probably, I mean, don't quote me on this, 10 engineers for every one scientist. Is it like that at Synthego too? Is it way more engineers working there than scientists?
I'd say it's becoming more of an even split. There's probably more engineers. I guess the way I kind of think about my title is a little more Star Trek-y, like, Spock was like the science officer, and you've got Scotty, the engineering officer. Anyway, going back to your question though. So yeah, my training, my undergraduate training was in molecular biology, and then my graduate training, master's and PhD, has been in microbiology, and but I actually worked. I think like most people who entered the CRISPR-Cas9 field, obviously back then, genome engineering was a whole different animal, if you will. And so it's something that I kind of fell into, mostly because after I left my PhD program at University of California, Davis, I went into working in a startup company that was actually trying to generate biofuels at the time. That was a George Church founded startup in San Francisco. And that essentially, when I was there, me and a couple other people, we sort of founded this synthetic biology group. That was kind of the sexy buzzword back in 2010, I guess.
Oh, yeah. That was like very Craig Venter at the time, right?
Exactly. So kind of, synthetic genomics and all that stuff. So kind of working in that field of synthetic biology where we're, trying to apply sort of engineering principles to how we build and design and sort of test biological systems. part of that is developing and adapting methodologies. And so it could be like a method that someone's published on. Maybe you want to adapt it into your lab, or maybe you want to combine methods or try to develop your own. But I had read the early CRISPR-Cas9 papers that came out from Jennifer Doudna's lab at UC Berkeley. This is back in like 2012, 2013. And I was starting to try to adapt those methods into my own work for this company, for the startup company. So that's kind of how I got involved in the CRISPR-Cas9 world of genome engineering. And, I tried to do some genome engineering when I was in graduate school. That was actually part of my research project, was to do genome engineering on a bacterial strain. But it was just so much harder back then. So looking back, it's one of those things where I wish I had this technology when I was doing my PhD, because I could have done it much faster.
Oh, for sure. I mean, that's kind of like the big takeaway sort of when we talk about CRISPR and we talk about sort of modern genomics is there was like a before and there was an after, And so that's sort of something that I would love to actually get into a little bit more, because I'm thinking we're throwing around a lot of terms on the show and sort of making the assumption, and I think it's a fair assumption based on my audience, that people have heard the word CRISPR before and that people kind of generally aren't too scared when they hear the word genetic engineering or genomics, but maybe they are, and maybe they don't know what any of this is. And it's probably good. There's so much to break down. So before we even get into CRISPR and before we even get into some of these new approaches and techniques, maybe we can talk about exactly what genetic engineering is, what genomics are, kind of what this whole field entails first and foremost, even before we get into synthetic biology, because that's a whole other animal.
Yeah, I mean, so when you think about what it means to do genetic engineering in a cell, essentially what you're doing is you're, all of us have DNA. Every cell has DNA in it. It's the code of life, right? Mm-hmm. So as human beings, we have a copy of a DNA from our mother and another copy from our father that are separated into chromosomes. And, entirely this is called your genome, right? So you have two pairs of 23 chromosomes. And so I think what's really important when we start talking about genetic engineering, genomics in general, is that you have to remember that each one of us, we're a very dynamic organism, and every living thing is very dynamic in the world. And so our genomes are no different. So just as, we can see our hair growing and then, hopefully not falling out, but our hair grows, it gets replaced. We get sunburn, we get freckles, our skin can turn darker. Our features change over time. And so that we can observe physically. And the same is true about genomes. And what's really critical to remember is that your genome or my genome or anyone's genome is not this sort of pristine thing that is, encased in glass in a museum or something like that. So genomes are changing all the time. The DNA is breaking, it's repairing itself. It's under attack from, sources like cosmic rays and radiation and the environment, things that can enter your cells. It could be a numerous amount of different types of chemicals or toxins in the environment that just occur naturally. And so, DNAs are under attack. They're also shifting around all the time. A large percentage of our genomes are actually made up of these, like what we call retroviral elements that at some point in time were actually viruses that incorporated themselves into our DNA. And so, these things can actually jump around and they move around. So your DNA is constantly shifting, it's moving, it's rearranging. It's to that end, though, it is still pretty organized and it can maintain and repair itself very effectively. And so I think when we start talking about humans, us manipulating genomes, we have to remember that they're always self-manipulating. And so if we come in and we tinker with a thing here or there, it's possible that we may kind of throw off the balance. But in general, we're not doing anything that's massively foreign or unique to this sort of almost like living entity, if you will, this genome. So, I always think okay, you're working, it's like a painting. And so maybe you come in and you make an extra brush stroke on this painting. And so, it's probably going to be very difficult to notice that, like, you're looking at a masterpiece that Monet painted and you come and you make an extra little brushstroke. It's still the masterpiece. It's just now it's got your signature on it.
Okay. And so when we are talking about doing this kind of engineering, like whether we're talking about students in a genetics lab, at a major university, or whether we're talking about industry, what kinds of, we'll get to the approach, I think, in a minute, but what kinds of reasons would people have to go in and start trying to understand and potentially change this dynamic genome of an organism? And often we're not talking about like engineering people, although that is a conversation to be had. Like many people are using CRISPR-Cas9 or Cas-X now with like standard laboratory organisms like Drosophila, fruit flies, or C. elegans, worms, and things like that. So what kinds of things are people interested in engineering?
Yeah, so there's a whole reason why you would want to manipulate genomes. And so this really kind of harkens back to really like a revolution that's occurred in biology, starting literally way back in the 1970s, and maybe slightly before that, which was our ability to actually manipulate DNA in vitro in the laboratory. And so this sort of we call it recombinant DNA revolution. So the ability to synthesize chemically strands of DNA, because each DNA base is essentially just a chemical, and these can be made industrially using synthesis type chemistry. And so the ability to synthesize like sequences of DNA and then kind of move them around in the laboratory was extremely powerful and changing to our entire world. So why would you want to do this type of thing? Well, you can do, at first, all of the genetic engineering was done in bacteria or yeast. And so, we've relied on bacteria and yeast for, thousands of years to generate food for us, for example, like yogurt and beer and things like this. So really the application of this technology for industrial microbiology production was, kind of really one of the first things that people looked into. So perhaps you have a strain of bacteria that you use to in your yogurt, and maybe that is susceptible to a disease itself. So bacteria are attacked by viruses, for example. There could be a way that you could engineer that bacteria essentially to become resistant to that virus. So when you're making yogurt, you don't waste hundreds of thousands of dollars because your whole culture dies and you have to kind of start over.
Yeah.
And it also makes your food safer. So, really people initially started manipulating DNA and using this recombinant DNA technology to study organisms as well. We can use it to, for example, you can delete a gene in a bacterial organism or a cell, and you can understand, how that gene is utilized in the cell, why it's important. So in order for us to assess gene function, to sequence the human genome, all these amazing things we've been able to do, leading up to you getting your 23andMe profile, is we needed to manipulate DNA. And so really that's kind of where the genomics revolution started, and it's kind of borne fruit all the way through to a lot of different applications. You mentioned Drosophila. So this is a model, or what we call a model organism. It's I won't say it's simple. It's relatively simple to study an organism that is smaller and has much fewer genes, and you can kind of start to map out pathways and networks of proteins and how they work. And so being able to kind of perturb different nodes in those systems can help you understand many different things, like neuronal development. How does a fruit fly brain develop? And there are analogies in how that develops to how a human brain develops, both from an evolutionary and developmental biology perspective, and also all the way through to disease. How does this brain become diseased, and how can we make a comparison, do sort of like a comparative study to what happens in a human, for example?
Yeah. I mean, that's obviously the way that sort of biomedical science has happened for as long as we've had biomedical science, which is you start at a, like you said, relatively, I like that you use that term, a relatively simple or organismal level, so that you can develop models and then you can start to tinker with those models. But eventually you have to move into translational research. And that is where kind of I don't know, I don't want to jump into this too soon without kind of having the foundation for understanding CRISPR, but that's sort of where I think people are getting slapped in the face with these news articles right now that a certain scientist, I shouldn't say certain scientists plural, but that a certain scientist sort of decided to skip a whole lot of steps and try and apply these things, not going through the standard ethical channels to human biology. So I want to get back to that later. But before I do, we've been talking about why you might want to manipulate the genome. I want to get into how one might manipulate the genome. And as you said, when you were in graduate school, there was no CRISPR. So how did you have to go through and try and do some of this stuff? And what is the difference now that we have this new technology?
Okay, yeah. So to be perfectly fair, CRISPR has been around for a really long time. It's been around for millions of years, probably in bacteria.
True.
We just didn't know about it. We just didn't know about it.
That's actually a really important point to make, I think. Like, we hadn't harnessed it. It's like fire. Yeah.
Exactly. So it's something that, that we discovered. It's analogous to, I did my PhD in microbiology, so I studied infectious diseases, and I studied what's considered to be an emerging infectious disease. And what we mean by that is that it's only recently been sort of identified. So we've identified like the causative agent of the disease. We've identified how it's transmitted into humans and through different vectors and things like this. So, diseases have always been there. I studied Lyme disease in my PhD. And so this was first identified in the 1970s in Old Lyme, Connecticut. That's why it's called Lyme disease. And really, it had always been there. We just never had made this association between the fact that ticks bite people and they could carry bacteria that sort of passes on this bacteria into humans. In fact, I don't know if maybe some of your listeners had seen, there was a really good Nova program a couple of years ago, and I'll just make a plug. Nova is definitely probably the best science program that's on television. But there was a Nova about an early human that they'd found in, I believe it was in the Alps, that was, thousands of years old, almost 10,000 years old, I think. And when they did analysis on him, they found, he was found in sort of like frozen, like almost like part of a glacier. They found that he'd had a lot of this tattooing around his joints. And when they did some further molecular testing and some DNA testing, they actually found that he'd been infected with Lyme disease. And so Lyme disease was around back then in Europe. The tattooing probably wasn't done like for fun like people do today, but it was probably done to try to relieve pressure and pain in joints. And that's what they believe. So
that's fascinating.
So CRISPR is the same way. It's always been there, present in bacteria. It's a form of immune system, adaptive immunity that bacteria have to defend themselves against viruses. And it's absolutely incredible when you think about like how amazing it is. And sometimes when you study bacteria, I'm a little biased because I study bacteria. People think, oh, they're very simple type organisms. Well, they're actually quite different because each one of our cells are highly specialized and they can only do maybe one thing. Like your liver cell, your hepatocyte can only be a liver, right? A bacterial cell is a single cell organism and it has to essentially do everything on its own in order to survive in the environment. So it's doing a lot of things. And for bacteria to have developed their own adaptive immune system where they essentially, maybe you guys have broken down CRISPR on your show before, but you've essentially taken a fragment of the enemy invading you, in this case a virus, into the bacteria, and you've taken some of its genetic material, you've stored it like a data recording in your own genetic material, so you can recognize it when it comes to attack again, and you can eradicate it. So that's a really powerful mechanism, and it's not very different to how our own immune systems have evolved in mammals to protect us from viruses and also bacteria. So going back originally to what you were saying, I didn't mean to go on a side note there, but
no, it's great. And I think we'll probably dig even a little bit deeper in a minute after you finish answering this core question back to kind of how CRISPR works, because we haven't done a deep dive on the show, and I think it's probably a good idea to do that.
Yeah, so I think, going back to what you were asking though, so CRISPR wasn't around as a technology, at least when I was in graduate school. So we were relying on more basic approaches to do genome engineering. And the main problem with that was, I was trying to engineer a bacterial strain. So specifically, I was working on Lyme disease, and I was very interested to know, we had this model for Lyme disease. Lyme disease can infect mice, it can infect ticks, and later humans and dogs. And so we wanted to understand what exactly were some of the genes doing in this bacteria that were allowing it to kind of develop this like pathogenic niche so it could survive in a host. That bacteria is very successful at surviving in hosts for a very long time, and it can actually evade the immune system. It's quite almost like until you study disease, you don't realize how almost perfect some of the designs and the engines are. I don't want to say designs, but some of the adaptations are. And so what was really interesting was, we identify some putative genes that we thought were interested in a particular aspect of the disease. And so you want to go in and you want to perturb those genes. Maybe if you remove those genes, is the bacteria still capable of infecting a host the same way, maintaining the disease state itself inside the host. And so in order to do that, we had to rely on, I would say, I won't say barbaric techniques, but much more blunt instruments. And so we couldn't go in and actually make, very specific changes or very easily. So, it's orders of magnitude difference between using CRISPR and the technologies we had before to do genome engineering. Whereas maybe in the past, if you wanted to make a specific change, you'd have to screen, hundreds of thousands of cells just to find the one that had the change you wanted. Now it's sometimes virtually every cell has the change you want. So that kind of jump has been just game changing in the way that the genome manipulation could occur. And this is because CRISPR has arrived. It's really going to push a lot of R&D much faster. It's a very powerful molecular biology tool. Probably, I would say I like to think about the technologies that have really changed molecular biology and how fast we can do research, which helps us to get to discoveries faster and therapies faster. And, there have been several, sort of like jumps.
Yeah, we think about PCR is a really big one.
The PCR technology, sequencing, the use of the ability to synthesize molecules, like I mentioned, DNA, these types of things. It's right up there with one of the formative steps. I think people will look back on this in some time in the future and say, hey, that was actually a landmark discovery that really changed how people could do
sure.
Genetic manipulation and analysis and do disease research and all these other applications you get from CRISPR.
And I think it's one of those things where we don't really even necessarily, obviously I think hindsight will be even stronger, but the actual practitioners who are doing this work right now are well aware of how much more accessible CRISPR has made this kind of work, both in terms of a speed and an exhaustion level, but also just in terms of a monetary level, labs that don't have quite the same funding or startups, kind of industry startups are able to do things that previously you could only do with massive NIH grants or with a lot of institutional support, which is really, really cool. But I think before we even get into sort of what synthetic biology is or how things have changed between CRISPR-Cas9, which when we say CRISPR, we're usually, or at least historically, we were referring to CRISPR-Cas9, but now CasX, I'd love to even just take a more fundamental approach to talk about what CRISPR is. So I had to Google this because, of course, I don't know it off the top of my head. But CRISPR stands for Clusters of Regularly Interspaced Short Palindromic Repeats.
Yeah.
So what we're actually talking about there is base pairs, like nucleotides? We're talking about little sections of DNA?
Essentially, yeah, we're talking about short, clustered, regularly interspaced. So what it means is like they're
it's just like just as it says.
Yeah, exactly. So, these are, you're correct though, these are short sequences of DNA, and they would be double-stranded DNA, so they'd be base paired, yeah. So they're short sections of sequences of DNA, that were regularly interspaced, so they were spaced at regular intervals in genomes. And so it was called CRISPR by the scientists that discovered this originally in bacteria. And so it was when they were starting to sequence bacterial genomes, because we started to develop the technology to do whole genome sequencing. People started looking in different bacteria, maybe the bacteria they were studying, for interesting motifs in the genome. And this was something that came up, that a lot of bacterial genomes had these like sequences that were like highly repeatable. And it's very strange to see those. Like, why would a bacteria sort of maintain this, what we called junk DNA at the time, just a bunch of like random repeats in a row?
So it's almost like looking for a pattern in the noise, like something that's like, oh, why is this doing it over and over and over and over again? It's very recognizable to like a software program. Right.
Yeah. So, it was sort of like, these are very strange. Why they here? What function and purpose do they have? There's really sort of like no reason to keep something around if you don't need it. And so there was a big reason for this, it turned out.
And that was?
Well, that was the fact that, so it actually turned out that the repeats that were common, they form one part of the CRISPR defense mechanism. So, if you will. So, what we call CRISPR, even though we call CRISPR-Cas9 or CasX, whichever one you want to talk about, Cas10, whatever, it's actually named for the region of the DNA that's not that interesting. So the repeat region is known as a tracer RNA. And so this is a standard piece of DNA that is later transcribed into RNA in the cell. And this piece basically acts as an anchor to anchor onto a protein, which is Cas9 or CasX, whatever one you're working with. And originally it was discovered in some of these organisms, the work that Jennifer Doudna's group did, where they showed that this repeat region, this tracer region, was binding to this Cas9 protein. At the time, they were studying Streptococcus pyogenes, which is the bacteria that causes strep throat. And so it turns out these repeat regions, they anchor onto a protein. What's more interesting is the regions that are in between the regularly interspaced sections. So in between the repeat regions are regions of unique DNA, and they're very short. And so typically, there were somewhere between 20 and 30 bases of DNA. And these were actually the memory, if you will. So these were the pieces of foreign DNA that had been incorporated in between these spacer regions. So these tracer regions. So, CRISPR is really named for the repeat section. It's probably like not the most interesting part, because it's not the part that actually does the business. What does the business is the piece that is immediately preceding that repeat region, which is known as a CRISPR RNA. And so this is DNA that is derived from an invading virus, or in the case of the bacteria. And the DNA gets sheared up from the virus when it's inside the cell. Sometimes the viruses die. Their DNA gets sheared up, and then these pieces will get put inside of these repeat regions by the bacterial cell, and it keeps it there as a piece of memory. And so when the cell gets infected again with a virus that contains that piece of DNA, invariably that virus will lose some DNA. Some of them will die and break up and open the DNA. And what these viruses are trying to do, are they're trying to incorporate their genomes into the bacterial genome so they can copy them many times over.
That's what viruses do, right? Like in any sort of organism, they sort of inject themselves into the cell and then multiply. And the cell actually takes in some of that DNA, becomes part of you.
And like I mentioned earlier, our genomes have lots of viral DNA, I would say ancient viral DNA in them. And some of them are still able to actually jump around and move around different places. It's just the nature of what DNA does. It can sort of homologously recombine with regions that are similar to itself. And that's sort of just the nature of the chemical in a way. And so, what was happening in this CRISPR system is that because what the bacterial cell will do, it would actually copy that repeat region along with that unique piece of DNA it's incorporated, and it would generate this into a piece of RNA. We call it a guide RNA, and that would guide that Cas9 nuclease, that protein. It's basically a piece of software code, if you will, that can tell that protein like where to go. It can direct a specific address on a genome. But that's not the way it works. The way it works is Cas9, the nuclease, the protein, we call it a nuclease because it's capable of cutting nucleic acid DNA. It's an enzyme that can break down DNA. What that protein actually does is it actually scans through an entire genome. So it's constantly interrogating a genome, and it's sort of coming up to a location and it's saying, do you match with this piece of code that I have that I'm carrying with me? No, you don't match. I'm not interested in you. I'm moving on. If it does match, it says, you match what I have carried here. I'm going to cut you, and then it will make a cut.
Mm-hmm. And this is why you often hear the term like molecular scissors. Like that's kind of what you do with CRISPRs. They're like little molecular scissors.
Exactly. So at this point, it's located a match for what it's carrying around with it. It's basically its code, if you will, that it's matched up to something. It's made a double-stranded cut. It's cut both strands of the DNA, and now the cell has to figure out what to do with itself. It has to repair itself. If it doesn't repair itself, it will die because these breaks can be very toxic because they essentially can screw up all of the coding regions and things like this in a genome.
Okay, so it's gone through it's gone through this whole process. It's done this. We're talking about kind of natural CRISPR at this point, like this happening in nature. How did we figure out how to harness it and actually use it for our own purposes? Like, where did that leap come through?
Yeah, I mean that's really kind of the genius point of this whole thing, right? So, this was identified by several different groups in bacteria. I would say probably the most notable of these was Francisco Mojica, who was at University of Alicante in Spain, and he was studying what we call halophiles, so bacteria that love to live in salty areas. And he'd been looking at genome sequences in these bacteria that he was studying, and found that they contain these repeat regions. And he was actually one of the first people to name this thing CRISPR. And I guess I should, before I go any further, I should kind of give a little bit of a plug. So there's a very good, very well written new scientific documentary film that premiered at South by Southwest. It's called Human Nature. And if you want to see the trailer, you can go to humannaturefilm.net. That's my movie plug. I'll be on the Tonight Show next week. Anyway, and so
wait, did you work on this film?
Yeah, so we were actually.
Is this your film?
No, it's not my film at all. So, this was a film that was directed by Adam Boltz, and he was part of Inside Job, which was a fantastic film. Dan Rather was one of the producers of this film, and along with several others, including Sarah Goodwin, who's part of this iBiology project that UCSF does. And essentially what they wanted to do was to make a documentary film that kind of showed not only kind of the history of CRISPR, but also how it was discovered and so forth, how it actually works, how it's being utilized, and also talk through a lot of the ethical questions around the use of this technology. So it's a fascinating film. My company, Synthego, was involved with the film a little bit. They actually came and did some filming at the company. I think I appear in the film for about 15 seconds. It's my 15 seconds of fame, literally. And,
Love it.
But it's narrated completely by all of the scientists who were involved in the development of this technology. So, for anybody, they're looking to get a wide release into cinemas for everyone to see. They've been doing some different showings. I went to the premiere at South by Southwest, but it's a fantastic film. And I guess what I'm trying to say is that it really does, for any of your listeners, it really does have some fantastic visualizations about how CRISPR works, and also documents the history of this amazing discovery and its different applications.
All right, guys, let's get back to the show. Well, and that's so helpful too. I think sometimes the podcast medium, allows us to do so much because we can really do these long form, deep dive, more nuanced interviews and talk about things in a way that you could never do with like your, minute and a half NPR interviews. But that said, it's an auditory medium. It's not a visual medium. And the truth is, you're right. In order to really understand this, I think we can understand it at the level of analogy and metaphor, and we can understand it from a practical perspective. But if you really want to understand it technically and like logistically, it does help to see graphics, to see animations, so that you can actually see what's physically happening inside of the cell down at the DNA level when CRISPR is doing its thing. So thank you for that. I think that's really helpful. One more time, what's the film called?
It's called Human Nature.
Human Nature. Okay, yeah. Very cool.
So I would say, for your listeners, they should definitely try to check it out when they get a chance.
Oh yeah, and for me, for sure. Like, this would be very helpful, because this is definitely one of those topics where, like I said, on my other podcast, we talk about it literally all the time. I'm always reading about it, and every time I have to give myself like a new primer on like how it works, because it's not my field. And I think for a lot of people who don't work in the sciences at all, it's like each time they have to remember, wait, what's going on now? Why is this happening? And I think there's a lot that we can sort of take for granted and say, okay, sure, going in, editing the genome, using this really cool tool that makes it a little bit easier and a little bit more precise. There's a lot of wonk there, but what happens once we edit the genome? So there is this portion where I think it's so important to understand the how of it, but the why of it is a whole other question. And the what happens next of it is a whole other question that is really, really important. And I think, not to transition too early, but that's really where synthetic biology comes in, because CRISPR and genetic engineering or gene editing is one thing. It's a technique. It's an approach. Synthetic biology is basically it's a whole new field, isn't it?
Yeah. So I think really depends on like what you define as genetic engineering, because essentially, and the film shows this, and it's something that's well known as well. But we've been manipulating genomes for a very long time as a species. So whether, now we're doing it in the laboratory, it's a little bit different, but we've actually been manipulating genomes for, literally tens of thousands of years by breeding plants together, breeding livestock together, utilizing microbes to make food, for example. So
yeah. And this is something that we do talk about a lot on Talk Nerdy, because we talk a lot about the GMO debates and things like that. And we talk about the difference between conventional breeding and transgenic technology, which all gets lumped together when we talk about GMOs, which is very confusing. But yeah, we have been manipulating sort of quote unquote natural selection and doing artificial selection for as long as we've been a species, and figured out, well, as long as we've been doing agriculture. Maybe when we were hunter-gatherers, we weren't doing it quite as much. But even with selective breeding of people, right? We chose to mate with people because of their traits. Right there, we started to manipulate the genome.
Exactly. Yep. And so, we're all kind of, I think as we go forward in time too, we're going to be doing this more and more. I'm sure one day people will say, hey, let me look at your 23andMe profile before, I want to go on a date with you. Right. So it's going to, we're going to be kind of selecting for traits that we want in some kind of way.
Yeah.
We already do that, and to some degree, we already do that.
Yeah, there are definitely people who know that they're carriers of certain genetic diseases that actually have to do these kinds of counselings before they get married or before they decide to have children. People with Huntington's disease or people who have like Tay-Sachs or things like that. They're actually pretty knowledgeable about this kind of stuff. But you're right. I think it's just going to become more commonplace in the future. Okay, but when we use terms like synthetic biology, because the company that you work for, Synthego, this is a Silicon Valley company. You went from academia into industry, which is a whole other thing that I feel like we could talk about for like an hour, because that's a big part of the show as well. Is like quote unquote alternative careers in the sciences, which aren't even alternative. Like it's just a career in the sciences. You don't have to stay in academia when you finish your PhD. If you finish your PhD, there's a lot of cool stuff that you can do out there. But so you're working in industry and you're working for a company, Synthego, that I take it from the name of the company is a synthetic biology company. What is A, synthetic biology, and B, how does that roll up into what you do at Synthego?
Yeah, I mean, if you sort of like take the definition of synthetic biology, and I'd say it's actually one of those things that people are still continuing to define. There's a synthetic biology conference, a large one that happens every year called SynBioBeta. That's put on in San Francisco, sometimes in London. And, I'd say at each one of these conferences, there's kind of like almost like a redefining of what exactly does it mean to do it. But in general, it's sort of taking almost an engineering approach to how you do biology, because you're actually trying to design and construct new biological sort of entities, if you will. And I don't necessarily mean living things, but new biological enzymes or genetic circuits or even cells, or maybe redesign existing biological systems. And you're trying to use almost like a standardization type approach to do that. And so that's kind of also ties into the company I work at, Synthego, because it really has a lot of engineering foundations and we could talk about this as a pretty interesting story at some point. But the two brothers that founded Synthego, Mike and Paul Dabrowski, they used to work for Elon Musk at SpaceX. They were pretty early employees there. And so they were really interested in understanding how they could apply engineering principles into the world of biological research. Because if it makes sense to me now, but before it didn't really, but it really does make sense that you have more engineers and scientists working together. Because if you look at what happens in a biological lab, there's still a lot of things that are done by hand. And unfortunately, humans make mistakes. And, I'm not suggesting that we have, an army of robots that does all the biology in the world and all the science and takes away all the intangibles. But sort of in general, there really does need to be sort of better levels of standardization in science. And part of the way we can see this indirectly is by how many publications are put out where people produce data that is actually not reproducible by other groups, or methods by other groups. So this is kind of a big problem in biology. People don't really want to talk about it too much, but if you can build levels of standardization into how you do biology, and in the case of the company I work for now, Synthego, how you actually do CRISPR-based genome engineering, it really can sort of move everything forward faster. And so it's gonna help us get to discoveries faster and therapies faster and things like this. Earlier you asked me a question about how did the actual formative change happen where people realized that you could utilize this CRISPR technology, this biology, if you will, CRISPR that was existing in cells and utilize it in an industrial way. There was kind of, there were kind of a couple of different moments that happened. One of them was, there was a scientist at Danisco, who, which is a yogurt company, which is making yogurt, who realized that they could use this as a way to protect their yogurt cultures. And that was kind of really the first, I would say, industrial application of this. And then came out the papers from Jennifer Doudna's lab and also Feng Zhang's lab at the Broad Institute. And, they were really the first ones to show that, hey, wait a minute, you've got a system here where you're actually directing a nuclease to go to a specific address on a genome and make a change. And that's what it nature does, and like, hey, you could actually, it's one of those oh shit moments, right? It's like you could actually use this as a really powerful tool in the laboratory. If you could take the kind of bare bones part of that system and you could get it to work in a lab, you could essentially manipulate any piece of DNA you wanted to, like in a specific manner. And so, those were definitely, I imagine at some point we're going to see a Nobel Prize for CRISPR, just because, it really was like one of those like, light bulb moments that, several groups had that really, really was like, oh shit, like we could use this this biology to do this.
Yeah.
Sort of like PCR, right? So
for sure. It's like such an interesting thing that it's a fundamental process that then has been harnessed in an effort to basically develop a technique that's foundational to almost everything in this field now. And it bypasses so much headache for so many people to be able to do this cheaply, easily. And probably it's much easier to train people to do it as well, right? Like you don't need nearly as much fundamental education to be able to like overcome potential mistakes. So
potentially, although I will always be an advocate for people to have, the right kind of training to use this type of technology, and the right kind of fundamental understanding.
For sure, but you don't need a PhD to be able to work in a CRISPR lab, do you?
No, I'd say you don't, but I'd say the same is true of anything like sequencing or PCR or any of these other technologies we've used. Routinely, people in the profession who just maybe have a bachelor's degree are doing this kind of work as well.
That's true. I guess lab engineers are generally not, or lab technicians, I apologize, are generally either still in school or they have, that basic degree that then they are able to, you're right, it's all about doing it over and over and over and having a lot of training in the area to develop that kind of expertise.
Yeah, so I'd say, one of the things you mentioned earlier in the interview was that, this is something that has kind of made it almost a level playing field where a lot of people can use the technology. And that's actually really a really important point. Is one of the great things about this technology is that most labs already have most of the equipment they need to do CRISPR experiments in their labs.
Oh, really?
So it doesn't require a new piece of capital equipment or a large investment to start doing CRISPR in your lab. So or for startups. So that is actually a really important point.
But, you hear these, and I guess this is the part where we're going to start like crushing headlines and things like that. But you hear these kind of fear-mongering stories, too, about the fact that now that this is such a democratized technology, and you're right, it does require training and it does require a lot of reading and understanding how to utilize it appropriately. But you do often hear that once these kinds of things are democratized, once they become cheaper, easier, faster, that then somebody in their basement will be able to utilize CRISPR technology and do all manner of horrible things. And I think that's really where the education is so important, both from a journalistic perspective, but also from an academic perspective, to sort of understand the realities and also the fear mongering. That it is true that people have access to technology that they maybe didn't have access to before because they simply, there's not a financial burden, but you still have to be like a scientifically minded person and really understand this kind of stuff and have a important hypothesis, like you have to have a purpose to be able to do that. Like, I don't think CRISPR in and of itself is causative for any sort of like nefarious practices.
Yeah, I think you're right. The other thing is that, like you mentioned, you need to have a particular degree of training in order to use this. I think what people need to understand is that the CRISPR technology is a tool that we can use for many different applications. We can use it for research. We can use it for to develop therapies, and it can also be used, for other purposes. But I think this is true of many different technologies. Whether we're talking about, computers, right? Computers can be used like we're using them now, or they can be used to hack into, everyone's bank account. So, I think whenever there's a new technology that's sort of transformative like this, I think there's always sort of like a segment of the population that sort of naturally questions, do we want to adopt this? And that's okay. That's part of like, the human dialogue that's part of, discourse. And so, I think what we have to be careful is that, in order to, a lot of people fear things when they don't really understand them, right? So, this really comes down to not only the scientific community, but also, I would say mass media, educating people the correct way about what this technology can and cannot do. And, so I think we've already seen a few like Hollywood films, where I think they had this film Rampage, right? Like based after like the 1980s arcade game where they made these giant animals and they're like, oh, we use CRISPR to make these animals. Okay. So that's not going to happen like anytime soon. And everyone starts to go back to like, everyone thinks of like the Jurassic Park model, like, there's a famous line in that film where I think, you didn't think about, what you could do, but like, what you would do with it or like what could happen. Right. And so, I don't think in general, like any scientists are like really thinking about like, oh, let's just do something for the sake of doing it. There's always a purpose to research. There's always a purpose to developing like a therapy, for example. And so I don't think, unless you got specific purposes to like, try to do harm with the technology, then it's it's really the responsibility of the people using it and also developing it to educate everybody about what it can and cannot do.
Absolutely. And I think that that's where these stories like the Chinese researcher and the quote unquote CRISPR babies really captured people's imagination, that this was a function. And I think in many ways it answers all of the things that you just said. This was a sort of kind of rogue researcher who was actually trying to solve a real problem and was actually trying to do quote unquote good with this and sort of didn't for several reasons that we could probably open up, but that's not what this episode is about. And fundamentally, the real issue there is that he decided to move forward with research that was not probably IRB approved. There were no ethics boards that were weighing in on whether or not this was something that the scientific community was ready to support. Basically, he went rogue. And that's really the problem, is that science doesn't work that way. And that's why there was an international outcry and backlash, because every science experiment that's done at an institutional or industry level has to be approved. There are mechanisms in place to ensure that the ethical sort of guidelines and boundaries are respected. And that's just something that didn't happen in this case, which is why it was such big news.
I think it wasn't just that. I think it's also the fact that a lot of scientists, we looked at what experiments were done, and we kind of realized like, hey, you know what? Even though they didn't have the necessary approvals, this actually wasn't done as a very good experiment. And what I mean by that is that like, this researcher tried to solve a particular problem, like, he didn't even actually target probably the right location on the genome to have the expected outcome.
And when he realized that he didn't have the expected outcome, he went ahead and did the implantation anyway.
Yeah, so,
which is, like, he was aware of it. He was aware that he probably didn't manipulate the appropriate gene. And also, again, he did it in people.
Yeah, so, I mean, obviously, I think this person is going to become ostracized from the scientific community. And, and it's good that there's a consensus among the scientific community that, like, what this particular exact experiment was unethical. I think in general though, like, some other people have come out and said like, hey, we should have a moratorium on doing this, on doing these types of things. It really, I think it really depends on if, we have to think about like what the needs are, where the technology is. And I think most of the people calling for a moratorium on this are saying we're talking about germline editing now, is that the technology really is not ready for that. And it's maybe not necessary because there are other ways that we can enact, we can be preventative when it comes to how we prevent diseases. Like in this case, this researcher was trying to prevent HIV infection. There are many different ways you can prevent HIV infection.
Yeah, it wasn't even the standard of care. Like there's functional, appropriate options available that don't require such a kind of dramatic approach. But just to be clear on the point that you just made, which is such an important one, germline editing is very different from editing somatic cells because germline cells are passed on to offspring. And so it's one thing to alter somebody's cells in their body that will affect their own lifespan and their own phenotype or disease state throughout their life. It's another thing to edit cells that then, if they mate, are going to continue to be passed down until that breeding line is no more. Because now all of a sudden you're affecting the population genetics, not just the individual genetics. So that's like a totally different ethical conversation.
Yeah, it's true. So, and I think that's why the first CRISPR-based therapeutics that we're seeing now, the first human trials have started in order to try to treat sickle cell anemia, which is a genetic disorder that's actually not that rare and affects millions of people on the planet and is quite debilitating, and is actually caused by a single DNA change to a single DNA base. And so there are groups that are working on developing therapeutics for this. There's a human trial that CRISPR Therapeutics and Vertex Pharmaceuticals are doing in both Germany and the US for sickle cell anemia and beta thalassemia, which is a related disease based on similar mutations in the similar gene, in the same gene. And, these are based on somatic cell editing. So, if you going back to the film Human Nature, one of the I would say the main stars in the film is Matthew Porteous, who's a MD, PhD clinician at Stanford University, and he's developing, and it follows some of his patients, and he's developing, a treatment for sickle cell anemia. Similar to some of these trials that are happening, and they'd like to start their trials soon. But he makes a really valid point, which is like, hey, we're doing somatic cell editing here. We're editing cells of patients who have a debilitating genetic disorder, and so to be able to improve their quality of life, we're doing this CRISPR gene engineering in some of their own cells in the lab, then expanding those corrected cells and then putting them back into the patient as a way to cure their disease. And so, the same will be true in the future of using this CRISPR technology to battle cancer in the case of what we call the CAR T therapies, where we can actually direct the immune system to destroy cancer cells. So from a somatic cell editing point, I don't think there's any real controversy about using this technology to do genome engineering in somatic cells. And I think when you go back to the germline engineering point, that's really where the controversy starts to come in. We have to admit, though, we do our own germline engineering all the time. And we've kind of have, whether it's through exposure to environmental toxins or, sort of selectively picking your mating partner or things like this. Those are all types of germline engineering, if you will. And I guess I'll just add on. We recently did a screening of the Human Nature film as well for all the employees at Synthego, and we had a few of the people who appeared in the film do like a panel at the end. And one of them is a gentleman named Hank Greeley, who's a professor of law at Stanford University, but kind of specializes in ethical and legal social issues. And one of the things he said was really profound, which was, so I don't want to take credit for this, but it was really profound. He said that, hey, we had a technology that had a lot of fear mongering associated with it about 75 years ago. It was the nuclear bomb.
Yeah.
And we decided as a population, as a species, that we were not going to use this technology. And even though we proliferated it for some time, we actually de-escalated that. And, up until this point, we've done some testing, but we actually haven't utilized this technology again. And so the same could be said of the CRISPR-Cas9 technology, right? Like we use nuclear technology to make power plants for satellites in space, or we use nuclear technology to for other reasons, right? For medicine, but the CRISPR can be the same way. We don't have to use it to do germline editing of people. We can use it for all these other applications that are really amazing and helpful.
Yeah, I mean, there's so much potential there, and so I guess that's an open place to start to transition a little bit into some of the things that you're doing at Synthego. Like what, that you can probably tell me, because I'm sure there's some like NDA kind of stuff going on there too, but what kinds of approaches are you guys taking? What kinds of problems are you trying to solve?
So I think, one of the first things the company did was actually to build a platform for actually synthesizing the RNA molecules that people use to do CRISPR. And this is because, that's because so it turns out the RNA is the way you program this nuclease. You can make these pieces of RNA in the lab, but they vary from who makes them and, the phase of the moon and things like this. If you could make these in kind of a standardized way using a chemical process, Jennifer Doudna's group showed that you could make a hybrid version of these RNA molecules that was slightly longer, different to what nature does, where it connects two pieces. So if you could make a single piece and synthesize as a chemical, it's really the most effective way to do CRISPR engineering. So the company built a platform for making those molecules, and it really took a lot of innovations in how we do that chemistry. And because the company has an engineering background, there was a lot of engineering and software applied to how we can actually operate machines that do this at a large scale. And so, this was really fundamental because it really opened up, it allowed researchers to access this new material that was really the most efficient and almost standardized way of doing CRISPR. And so that was really important. And then, beyond that, the company sort of layered that out into different levels of abstractions because, if it built a factory that could generate these molecules that could enable people to do a lot of CRISPR. We started to do a lot of CRISPR ourselves. We started thinking about how we could automate a lot of these workflows. And then we thought, hey, we can actually use this to kind of build another factory that could generate mutations in cells that people use to study disease and so forth. So what we call cell lines, so these are immortalized cells that grow in a dish. How can you engineer these at scale using this kind of a standardized approach? You're starting with a standardized reagent, and maybe you can layer some automation and some almost like machine learning to understand the best way to engineer cell lines at scale. So you can kind of build a more standardized group of cells, if you will, that people will use to study, to do their disease research, for example. And so that was kind of the next abstraction of the company. And then, from there, you start thinking about other things. And, I think the company will do other things as well. Like, how can you actually use this to build pipelines for discovery, or how can you use this to do functional genomic screening? If you think as a company right now, you sell somebody a reagent to do CRISPR, then you can actually do the CRISPR for them more efficiently than they can do. You can essentially, you have all the methods figured out. They don't have to waste time developing their methods. You've kind of standardized that, and they can spend more time thinking about their experiments than their actual method development. And then if you do that, maybe the next abstraction is also, you can actually just give them a piece of data. They can say, hey, I want to perturb this, I want to generate this mutation in this cell line, and then I want to run this assay and understand what it means. And so really, I think kind of the future of synthetic biology and biology is actually going to be that, where people, whether it's large pharma or research labs, they'll actually, what they really need is data. And so where they get that data from isn't necessarily incredibly important. A lot of scientists obviously want to do things their own way with their own hands, and that's fine. But I think if we can help to build standardization through platforms like this, it's really beneficial to the research community.
Oh, absolutely. So, what I'm gathering from what you're saying is that at Synthego, as opposed to answering these more narrowly focused and more, translational research questions, you're really focusing on providing the scaffolding so that to empower scientists all over the world to be able to do that. This is much more of a foundational approach to being able to solve some of these problems, because there's just so many potential utilizations of this technology. So A, you're making it easier for people to use this technology, and B, in the future, it may be that the actual methodology portion of the research is just farmed out to organizations that do this all day, every day, and they do it really, really well.
Yeah, that's kind of the direction, as a company that we're headed in. And, I think the other thing that's great is, too, we also do a lot of education, so you can check out our website if you want to. I don't want to give another plug, but if you check out synthego.com, you can learn a lot about CRISPR there. So we try to be very interactive with the community. We do a lot of interviews with people as well. We also do our own podcast. And, so we're also trying to educate the scientific community and people who want to, start using this technology, kind of what are like best practices, best methods, so they can, really, get to their results much faster and efficiently.
It's very cool. So, I mean, I'm definitely interested to know from you, like, what are kind of the next steps, like where, for you personally, but also for kind of your role in the company, like, what does the future look like for you?
That's always difficult to say. Sometimes people say, hey, Kevin, what do you see yourself in 10 years? And I always say, sitting on a beach, doing nothing, because I've accomplished everything I wanted to. That's a really good question. I think for me, at least, I'm definitely excited to see this company grow and develop into like a platform technology that can be utilized for many different applications. I see the company and myself maybe growing into a space where we're interested in many different aspects of utilizing this technology, whether it's for doing development of cells for people, or working with other researchers or clinicians to develop this material that can actually, in a grade that can actually be used therapeutically in a clinical trial. To me, that's really exciting because it kind of offers you the translation that every scientist wants. That, hey, I'm doing something that's helping humanity, and not only that, but also, how can we, how can we actually translate what we developed at this company into something that could actually, help humans from a therapeutic standpoint. So I think seeing this company grow from where it has, and it's a relatively new company. We've only been in the public eye for about 3 years. It's been a really exciting process to see the company grow, and this technology is just exploding everywhere, right? CRISPR-Cas9. So it's really exciting to see what's going to happen next. I definitely want to stay in this field. It moves very quickly. There are new discoveries being made all the time, and it's difficult to keep up, but it really is an exciting time to be a scientist, particularly in this field. So it's where I want to stay right now.
Absolutely. Well, I would love to then, sort of as we round out the interview and get ready to say our goodbyes, I would love to close by asking you the same two questions that I ask all of my guests on the show, especially because I think that they, I'm just interested to see if they're connected to your work in this field or if they're completely separate. I think both of those things would be fascinating to me. They're kind of big picture, future-oriented questions. Are you ready for them?
Sure.
All right. So when you think about the future in whatever context is relevant to you right now, so it could be, your own life, your own work, it could be the company, it could be something much more global and connected. I want to know, A, what is the thing that actually keeps you up at night that you're sort of maybe pessimistic about, maybe even a little cynical about? Like, what are you truly deeply concerned about when it comes to the future? And on the flip side of that, what do you have genuine, actual optimism for? What are you actually looking forward to?
Okay, I'm going to try to answer this in like the least political way possible.
It's totally fine. I mean, it's up to you if you want to do that, but like, no barriers here.
Okay, so I think what keeps me up at night sometimes is, and this in a way does relate to the field that I work in and technology in general. I think what keeps me up at night a little bit is, and what concerns me deeply, is that sometimes I get the impression that education is not valued among our species. And what I mean by this is, I guess I was sort of like, I grew up watching Star Trek, the original one, and also, the Next Generation, all these Star Trek things. I loved all of them, but kind of this like vision of the vision of the future where humanity at least has learned to work together. And, I think we can all see this like individually when we work at a company. I work at a company, and we can see that like when people come together as companies, we can do amazing things, right? And, you think about all the things that have happened in the last 200 years, the Industrial Revolution, getting people to like work together, and, hopefully we don't exploit people along the way, but like doing all these things, getting people to work together and use their brains together, we can do incredible things. We can manipulate our world. And now we can also manipulate genomes. So I think what keeps me up at night is that, there are large numbers of people still in this world that, and even in our own culture, whether it's Western culture or wherever that, essentially they don't pay a lot of attention to things like this. They just tend to read headlines, and they don't really want to deeply understand how things work. And part of this is because they haven't developed critical thinking skills. And so I think what keeps me up at night is that there is still a lot of ignorance in our world, particularly when it comes to science. And this leads to things like fear mongering, and this leads to people passing bills to like inhibit research or cut funding or things like this. And then I think sometimes you kind of see like this sort of rise of ignorance. From particular segments in the population, and then it kind of quells down again, and then it seems to like rise up again. I think, though, my optimism is that it's slowly getting better all the time. And what I mean by that is sometimes progress can be slow, but I do think eventually humans will learn to stop, doing harm to each other, and humans will start to think about ways that we can all be nice to each other, and that we can be much more productive as a species. And because there's some significant challenges coming, right? There's significant challenges in what's going to happen to our climate. There's significant challenges in terms of like what's going to happen with food, plastics in the ocean, like all of these things. And it's really going to take a group effort, and everyone needs to kind of be on the same page. And, if we're not, and you still got people who are questioning, why are we doing this? It really takes away from our ability to solve those problems and to be more successful as a species. So, but I do think there is the sort of overarching trend towards getting better. And I think for every time we see some like large amount of ignorance occur, there are other groups of people that come forth and try to educate. And I think slowly it sort of leaks in, and opinions change, and people's mindsets change. Now I think Burger King just announced they are using the Impossible Burger, right, in their Whopper sandwich. And honestly, like 10 years ago, people would just be absolutely disgusted and grossed out. I don't want to eat a burger that's not made from a cow. And now we're talking about like a hamburger that tastes like a hamburger and comes from a renewable source, and that's amazing. Like, so people's attitudes have changed, and and that could do a lot to solve a lot of problems. So I'm hopeful that attitudes change, and that people will become more educated. I'm not saying everyone has to be a scientist, no, everyone has to understand science, but I think they need to have an appreciation for it.
Yeah, I mean, I think a basic level of literacy is important in an effort to be like a participating kind of citizen. And you're right, like for every outcropping that we see of like anti-vax hysteria and individuals, having these small, not as small as we'd like, but semi-contained measles outbreaks and things like that, we always do have to remember that there are massive organizations like the Centers for Disease Control, and like these pharmaceutical companies, and like these academic and institutional organizations that are actively developing new vaccines for Ebola and for a lot of infectious diseases. Like we sometimes get, I think, a little bit depressed because we see that individuals who maybe aren't in the positions of don't have the basic education and are beholden to like some rhetoric, making bad decisions. And we think that colors the entire world. But it's true that while that exists, and there's always going to be this kind of anti-science segment, that is not actually in and of itself preventing scientific process. And I think we have to remember that, because sometimes the best science is happening maybe a little bit behind the scenes, and we're not as privy to the really cool stuff that happens because it's so iterative that it doesn't make for a great headline. And so we have to remember as individuals that there's so much important progress that's being made. And those are very different people, the people making that progress, people like yourself, as opposed to somebody who's like just an out loud, outspoken anti-vaxxer. Those things can exist within the same global society, and we can still make progress even when there are people who are, desperately trying to prevent it, I think. Gosh, I just went on a little tear. I apologize for that.
That's okay.
Been on my mind a lot lately, So Kevin, gosh, this was a fascinating conversation. I have to be honest, I feel like we could do a part 2 and a part 3 and a part 4 because we could just dive deeper into all of these different topics. Well, of course you could. This is what careers are made of. There's no way to cover it all within an hour podcast. But I do thank you for helping, at least in terms of a fair amount of breadth and a bit of depth as well, helping us understand CRISPR at a more basic level and sort of the potential that it has to do a lot of good in this world. So thanks so much for joining me today and teaching me something new.
Yeah, you're very welcome. Thanks for having me on, and let me know when you're doing 3. I'm ready.
Awesome. And everybody listening, thank you for coming back week after week. I'm really looking forward to the next time we all get together to talk nerdy.